is a base that will then add to the activated carbonyl. So this reaction is called a cyanohydrin formation. So practically speaking, when it's implemented, hydrogen cyanide itself is a poison. So that's not used as a reagent. So the conjugate base of that is used under basic conditions. So sodium bicarbonate with a biphasic aqueous alcohol mixture. So in the example shown, we have benzaldehyde. So our oxidation state is shown in 2. And what happens is we have an sp hybridized carbanion. So you can use any spectator here. So sodium and potassium cyanides are the commercially available ones. And so we know the carbonyl is delta minus, the carbon's delta plus, negative is attracted to positive, so that's the nucleophilic addition. The pi bond breaks, so those electrons go on to oxygen. And then we have this resting intermediate. That will then get protonated on workup. The other thing I want to point out, the carbonyl carbon's sp2, it's now going down to, or going up to sp3. Because that's not the final product, another way to refer to that is a tetrahedral intermediate. And that has relevance to biochemical mechanisms where people study transformations and sort of the stability of those type of intermediates. Can you trap them and isolate them, or do they break down into something else? So overall, what we end up with, the carbon's been reduced to oxidation state 1, but we've introduced a carbon that's at oxidation state 3. So a nitrile is functionally equivalent to a carboxylic acid because that carbon is also at oxidation state 3. So a nitrile to an acid is a conversion that's commonly employed. And if you think about the spectroscopic signatures of a nitrile, say in the IR, so in that bonus spectroscopy problem, the nitrile was one of the functional groups, and so that was seen in region 2 of the IR. So that has its own thing with an sp hybridized carbon. And then if you recall what a carboxylic acid looks like in an IR, they're very different. But the carbon is retaining that OS3. I kind of already showed the mechanism of this on the prior slide, so we'll skip that. And I want to elaborate a little bit more on, I don't think I have it written on here, so I'll say FGT, that's functional group transformation. So a cyanohydrin, that's a carbon that's bearing both the alcohol and the nitrile. When treated under really strongly acidic, so sulfuric acid or sodium hydroxide, either one. So either acidic or basic conditions at pretty high temperatures will hydrolyze the nitrile to an acid. And so that then produces an alpha hydroxy acid. Again, that alpha carbon is the one right next door to a carbonyl. So an alpha hydroxy acid is difunctional. We have a carboxylic acid with a pK of around 5, and then this alcohol with a pK of around 16 to 18. So those OH groups are differentiated by pKa. So even though I say on your own, I think it's important I'll walk you through this so that you understand the importance of proton transfers and, in consideration of the pH of the medium. So we're under acidic conditions, meaning that we can only have intermediates that are neutral or positively charged. One of the challenges of this mechanism, it's a lot quite steppy in terms of these proton transfers, but what's changing? We have to get that nitrogen out of the molecule. So how do we do that? What does it leave as? So we'll apply our principles here where we know that sulfuric acid is an acid. And based on what we're trying to do is get rid of that nitrogen. We see the nitrogen of the cyano group has a lone pair, so that can be a base. So we can protonate that. So I'm just going to write the acid as H+. So we have a protonated nitrile. We can draw a resonance form of that will help us understand how we start getting oxygens in there. So if we move a pi bond pair of electrons onto nitrogen, it forms this type of carbocation. This is a vinylic carbocation. Meaning that the positive charge resides in an sp2 hybrid orbital. We're normally used to seeing the plus charge reside in a p orbital. But regardless, that now is electrophilic. We're in the presence of water, which is a nucleophile. So that's the source of oxygen, and its lone pair can attack. And now that oxygen has the plus charge. So we'll do a proton transfer with water. So we want to make that oxygen neutral. So the thing that happens next is not resonance. It's a different sort of concept that's going to become important kind of in the next module. But if we see this hydrogen and we count the relationship between the heteroatoms, 1, 2, 3, this hydrogen can move over to that nitrogen. And that process is called tautomerism. So the next thing I draw is going to be a tautomer of this. This functional group may not look familiar, but the next one should. So when this H migrates over to there, so what's the name of that functional group? So the carbonyl with the nitrogen, that's based on these hydrogens, that's a primary amide. So what we've done so far is we got one of the oxygens in, we've kind of activated that nitrogen by putting some hydrogens on it, and ultimately we're trying to transform it into a good leaving group. So I think maybe what I want to do in terms of the arrow pushing, this is showing us kind of a way that we got the oxygen and then this NH2. But I want to branch off from here so I don't start putting negative charges in the arrow pushing here. So I'll explain what I mean by that in a minute. That nitrogen still has its lone pair. So at this point, let's use it as a base and protonate it. So when going up this way, I've done this proton transfer, so we still have this OH now. And then I've protonated that nitrogen. The reason I wanted to do that is to draw a resonance form from this, where we can get the second oxygen atom in. So this is going to get a bit squished here. So I'm going to move that up. Break this CN double bond. Oop. Should be, it doesn't like that. It should be resonance. Just to save some space, I'm going to abbreviate the ring as pH phenyl. So that will allow us to have another molecule of water to attack to get the second oxygen in. Starting to look like a bloodbath up there. So what do we have to do now? We have to remove a hydrogen from this. This nitrogen has to be protonated. So I'm going to show both of that kind of in one step, where water abstracts that, and then that gets protonated. So that would be protonation of the nitrogen. This is proton transfer from that oxygen. And now the nitrogen is positively charged. So what that allows us to do is either one of these remaining OH groups can reform a carbonyl and then eliminate NH3 as a leaving group. And so then the nitrogen leaves as a neutral molecule. So sorry for the messiness here. So we're going off the right-hand side, reappearing on the left-hand side. And then a final proton transfer. Will result in the carboxylic acid. So this is one of the, I would say, more drawn out mechanisms, because you have to do these proton transfers that jive with the pH of the medium. Things have to be neutral or plus charged. If you try to have nitrogen negatively charged, that's not going to be correct due to the sulfuric acid being present. So one thing, if you're drawing out mechanisms in this class, but as it translates into the biochem courses, the proton transfers are done with amino acids, which we're going to introduce in a minute here. So in enzymes, which are polymers of different amino acids in a peptide chain, you have side groups that are acidic or basic, and they're what is accomplishing the movement of these protons in a chemical transformation. Any questions? Give you a minute to give your hand a break as you stretch out the muscles. The other important thing I want to sort of note about this, the reason this is a powerful transformation, as we'll see in the next case where it gets a little bit more elaborate, the cyanide reagent, that carbon's negative. So there's this natural polarity match between the negatively charged carbon and the positively charged carbonyl to carry that out pretty easy. You can't really take a carbonyl compound and add a carboxylic acid functional group to it, because in the acid, based on resonance, that carbon's delta plus as well. So there's no reagent that has a nucleophilic sort of carboxyl carbon of an acid. So you have to go through this process to arrive at that specific arrangement. So we can extend that alpha hydroxy, the cyanohydrin, into the alpha hydroxy acid as a strategy into making alpha aminonitriles. And the reason that's important is because we said the nitrile is equivalent to an acid. And so this product that's shown is equivalent to an amino acid. And those are the building blocks for peptides and proteins. The name of this reaction is called the Strecker. So the Strecker reaction. So it starts with OS2, either an aldehyde or a ketone. We're going to use the cyanide, but notice now the difference in the solvent here. We have ammonia. So rather than the cyanide adding first, what happens is the ammonia condenses with the carbonyl. That results in the formation of a carbon-nitrogen double bond. That's shown in the intermediate here. That functionality is called an imine. So the CN double bond, which we haven't seen a lot of, we've mainly just dealt with carbonyls, is an imine. So an imine is the oxidation state 2 nitrogen variant of a carbonyl. So as with the prior process, we still have that same, the heteroatoms delta minus, the carbonyl carbons delta plus. So the cyanide can attack that, break the pi bond open, and then upon workup, we have the alpha amino nitrile. And as I said, in what we just went through, the hydrolysis of that nitrile can result in the formation of an acid. And so alpha amino acids, again, are the building blocks for biopolymers. And I'll talk about, their general structural features. The alpha carbon is going to be at oxidation state 1 with an amine. Then you have this acid. And then the side chain is what differs in amino acids. So everyone in this room is composed of these 20 proteinogenic amino acids. And all the amino acids, as I said, have that, but they differ by what this side chain is. So you can use this Strecker reaction to make virtually any amino acid, whether it's natural or unnatural, based on the starting aldehyde. So see the side chain here? Nothing happens overall, but it's what gives it the unique properties of that alpha amino acid. So I want to talk about imine formation, because that's going to lead to, as I said, another way to synthesize amines. And overall, the carbonyl to imine, you're retaining that OS2. And this is called a condensation. So we conserve that. So we have carbonyl going to imine. If we look at what is the other product of this, so we have NH3 here, we have an oxygen there. The product has NH, so what is the difference? It's producing water, so it's a small neutral molecule, it's stable. So condensation reactions, one way to think about them is they're producing this byproduct that wants to exist, that increases the entropy. One thing we talked about in Organic 1 is affecting the thermodynamics of a reaction. So using the delta minus, delta plus, we're going to show the ammonia attacking that. We break the pi bond. So this mechanism, I don't want you to think I'm lying to you because I'm showing both a positive and a negative charge in there. Based on pKa arguments, we can do this because we're not really under strongly acidic or basic conditions. So I'm just going to say proton transfers there. The O minus gets protonated, the N plus gets deprotonated. And then look at this carbon right here. What hybridization is that carbon now? So I'll draw that H in. So it's sp3 hybridized. And then what oxidation state is it? So it was sigma bond to nitrogen, sigma bond to oxygen, so it's still 2. Remember from the prior lecture when we had a sp3 hybridized carbon, oxidation state 2, but both heteroatoms were oxygen. Those were called acetals. When you have something that's mixed, nitrogen and oxygen, those are called aminals. So notice the difference between the M and the N. It's not an animal, it's an aminal. And so what we can do now is just an elimination where we get rid of H2O through proton transfers. So in blue, I'll just put the oxidation state number as we go through. And so now we have this imine, and that's the carbonyl, the nitrogen variant of a carbonyl. So it undergoes all these same reactions that you've learned so far in both organic 1 and 2. So because that heteroatom is now nitrogen, it can still undergo the Strecker by adding cyanide to it, and then ultimately you get the alpha aminonitrile. Any questions? We're going to go a little bit more in depth into the imines, but since this does relate to the alpha amino acids, I just want to briefly introduce them and some nomenclature about them. So, as I said, we know what a carboxylic acid is, we know what an amine is. The alpha amino acid, again, those are the monomers that we can start making peptides from, which we'll see in Module 5. And then this R1, again, that was the side chain. So really, they only differ by the nature of what that side chain is. And then as we'll see over the next couple of slides, there's 4 major categories of those side chains. Based on the stereochemistry of naturally occurring amino acids, that stereogenic center ends up being S when you apply the priority assignments to them, when you're doing the Condon-Gall-Prelog rules. When it comes to these amino acids, and then we'll see a little bit later on for carbohydrates, another nomenclature designator that's used is this. So capital L or capital D. And that relates to a Fisher projection, which we talked about in Organic 1, when we talked about stereochemistry. A Fisher projection is a way of looking at a carbon backbone from a different viewpoint. So in the left structure, the carbon backbone is right in the plane of the whiteboard. For a Fisher projection, you're kind of putting your eyeball like right here. And then the carbon backbone ends up being vertical. So that's now your carbon backbone. The tricky thing about this is, see how you have the vertical line, the horizontal line, they cross. What this translates to in if you build a model with your model kit, the horizontal line, those substituents are coming out towards you, so they want to hug you. The vertical stuff is going away from you. So the stereochemical information with the wedges and dashes has been sort of reduced into this form of looking at it through the Fisher projection. Since that carbon is a stereocenter, and the substituent with the most importance that makes it a stereocenter is on the left, it's given this designation L, which corresponds to the S stereochemistry. If the amino group was on the right, then it would be called a D amino acid, and it would have the R configuration. So unless your parents were superheroes from the Marvel universe and you have some sort of weird genetics, pretty much everyone has these L amino acids in their proteins. So based on what that side chain R is, there's 4 classes of amino acids: nonpolar, polar, acidic, and basic. So what I'm going to do today is just briefly show you what the side chains are, and then when we get to the last module, we'll see why the side chain is important for biological activity. So for nonpolar amino acids, basically, you can think of this as kind of 2 different ways, either lipophilic or hydrophobic, depending on what kind of medium you're in. So lipophilic means fat-loving, hydrophobic means water-hating, the same way of describing things. But basically the side chain's kind of your normal CH stuff. So glycine, notice it does not have a stereogenic center. The alpha carbon is just 2 hydrogens. So these are the names: glycine. They have 3-letter abbreviations, so GLY. And then they're further abbreviated G. So has anyone taken cellular or molecular biology courses yet? So have you seen the sequences of like proteins? Looks like someone's just randomly typing stuff on a computer. It's just like a string. That string of letters is corresponding to an amino acid. So that, however many there are, it's just a long chain of amino acids. And then based on these functional groups present in the side chain, but not only how the amino acids are bonded, you'll get these secondary and tertiary structures that will arise from that dictate the biological activity. So for alanine, there's a methyl. Valine, it's isopropyl. Leucine, you have this isobutyl. Notice isoleucine has a second stereocenter. Methionine has a sulfur, so that's present in a sulfide, so the sulfur variant of an ether. Proline is the only one with a ring. So notice these other ones, that amino group is primary. Proline's the only one that's a secondary amine. Phenylalanine and then tryptophan. So you have an aromatic ring, and now you have a heteroaromatic system. So this thing here, benzene plus that, is called indole, and that makes tryptophan. The polar amino acids have some other functionality. So asparagine has an amide over here. Glutamine is another amide, but there's a longer linker from the alpha carbon. Serine has an alcohol, a primary alcohol. Threonine is a secondary alcohol with another stereogenic center. Tyrosine is like a variant of phenylalanine, but now you have a phenolic group, so an aromatic alcohol. And then cysteine is this thiol version of serine. And notice that changes to R just because of the priority of that sulfur. So cysteine is the only one with the R stereochemistry. In the Fisher projection, that amine group still resides to the left, but just due to priority from atomic number, it switches to R. Acidic amino acids, aspartic acid and glutamic acid, both have a second carboxylic acid, and they just differ by the number of CH2 groups. So 1 to 2 from the alpha carbon. Basic arginine. This group here is called a guanidine. So you can think of that as you have a second amine, and that amine is bonded to this part. So that whole thing is called a guanidine. Histidine has an imidazole. So that 5-membered ring with 2 nitrogens is called imidazole, and we spent some time talking about basicity of nitrogen lone pairs. So that's the relevance of that concept to this. So the lone pairs reside in different orbitals. So in histidine, the lone pair in the sp2 hybrid orbital is basic. And then for protein function, that can act as a base. So that helps shuffle protons around, like in the mechanisms we were talking about prior. And then finally, lysine has a second primary amine. So you have a primary amine at the alpha position, then you have one at the end of the chain. So if you're going into, one of these more focused biology majors, that's cellular or molecular biology, you'll kind of have to know these by heart, because they're really ultimately reduced to these single letters. And you do want to have an idea of what that letter is informing you about on the structural scale. So let's circle back to imines, because those were the intermediates for the Strecker. And I want to talk about the condensations with the different oxidation state 2 functional groups versus the class of amine that you're starting with. So imines have a carbon-nitrogen pi bond. So in blue, I'm just going to record the oxidation state. So 2, 2, 2, and 2. And then with a red line, I'm going to separate between those 2 sets. The difference between them, these are derived from aldehydes, and these are derived from ketones. So how do we know that? We've identified the sp2 hybridized carbon, then we look at what's attached to it. So carbon, hydrogen, that comes from an aldehyde. Over here, carbon and carbon, that comes from a ketone. But now let's dissect it in a different way. So I'll use the green. So imines are the product of a condensation between an aldehyde or a ketone with a primary amine. So when I do that dissection, if you think of that coming from a carbonyl, what amine is it condensing with? So remember, the overall process is a condensation, which loses H2O. So this first one has been condensed with ammonia. This second one has been condensed with methylamine. So the difference is, see the H versus that? Nothing happens there, but the nitrogen has to be primary. Because we're losing the oxygen and then those two hydrogens. And then we make the CN double bond. So like on the exams, if you're given the structure of an imine, can you determine what carbonyl functionality has been used, an aldehyde or a ketone, and then what amine has been used? So we kind of did this already, but I'll dissect it again. So I want to dissect the double bond. That carbon was oxidation state 2, so it has to come from a carbonyl. The fact that we have a carbon and an H means that the carbonyl belongs to an aldehyde. So that would eliminate C and D. And then when we look at what's up here, the substituent remaining on nitrogen is hydrogen. So that means we have to start with ammonia. So that's going to eliminate B. So ammonia plus butanal is going to give you that imine. So the important thing to the pair to remember is amines come from primary amines. If you start with a secondary amine, this generates a different functional group called an enamine. So that's a mashup of an alkene plus an amine. So why is there a difference between a primary and a secondary amine going to different things? Again, that relates back to you have to produce water, H2O. Now, if I dissect that bond, we see that the nitrogen has 2 carbon substituents. So it has to come from a secondary amine that's condensing with the carbonyl, because we're using that as oxidation state 2. So we're trying to produce water. So if we count, we have one H, we have an O. So H and O is not H2O. So where does the second H come from? It has to come from the alpha carbon. And so that's how we end up with now the double bond is between the carbonyl carbon, the alpha carbon, to get the alkene. And then you have the amine part, so that's called an enamine. So looking at any enamine, you should be able to determine, is it coming from an aldehyde or a ketone, and then what secondary amine has been used. The two structures shown, because you have a double bond, you can assign E or Z to indicate the cis or trans stereochemistry. So let me redraw, because I'm getting a rainbow of colors over here. So this one was said to be E, and how do we arrive at that? We identify both carbons of the alkene, look at what's bonded to it, and assign priority. And the priority is based on atomic number. So we have N versus H. So N gets priority 1, H gets priority 2. Now we do the other side. We have carbon versus H. So carbon is 1, hydrogen's 2. The ones are opposite, so that would be E, which is another way of saying trans. Any questions? Let me just point out, see this, see the aldehyde over here? The aldehyde only has one alpha carbon. So the elimination to form the alkene can only go one way, but it can result in cis versus trans. So ketones are a little bit more challenging because ketones, on the other hand, because both things are carbon, the double bond can form either way. So if we do the same dissection, there's the oxidation state. Now the starting carbonyl is this ketone, so there's an alpha carbon and then an alpha prime. So there's two alpha carbons. They're different because one's a methyl and one's methylene. The amine is still secondary. So that secondary amine condensing with this, the final H of H2O can either come from alpha or alpha prime. So in the first case, we've eliminated from that alpha. The second two was from that alpha prime, and because we have these different substituents, we can get the E versus Z geometrical isomers. So I have this question: can you think of why tertiary amines cannot be used? So methylamine is primary, Dimethylamine is secondary. Trimethylamine is tertiary. So a tertiary amine will not condense with an aldehyde or a ketone. So why do you think that is? Remember, if the goal is to produce water as the condensation byproduct, the tertiary amine no longer has a hydrogen bonded to nitrogen. So the most you could get out of this is an oxygen and then a hydrogen from an alpha carbon. So you could only produce OH, not H2O. So we can use that to our benefit. And then really the take-home message is whether you're given an imine or an enamine, can you identify the carbonyl and then what class of amine has been used? So I'll push the arrows for this, and I want to do this under acidic conditions so I can show, some proton transfers, and then really when we get to that point of why the alpha carbon CH bond is invoked in this. So with proton transfers, since they happen pretty fast, we have an amine which is a base in the presence of acid. So the amine can get protonated. So that can happen on and off. But also the carbonyl carbon can be protonated, so that activates it. So let's start with that because it's productive. So we'll protonate with hydronium. And now we have this activated carbonyl in which we can show a resonance form where that carbon is positive. And that's going to allow us to have the secondary amine attack that. So now the plus charge is on nitrogen. So I'll show two proton transfers at once here because we got to get the OH out of there. We got to get the H off the nitrogen. So here's our standard flux capacitor acting as the acid. We'll have water be the base down here to do that proton transfer. And I'm gonna, again, screw myself by running out of room. So we've set the stage for the lone pair on nitrogen now to collapse down. That will form a carbon-nitrogen pi bond. So it's going to eliminate out water. Why am I drawing H's there? Sorry. This should be CH3. It's the whole point of this. So when we do that, that's what we end up. That nitrogen is now protonated, or not protonated, it has a plus charge, which is now different from the imine case where the nitrogen had its lone pair. So it wants to get back to that situation where it has the lone pair. So we had alpha prime and alpha. And so the last acid-base reaction, I'll just show one of them. We're going to deprotonate, form the alkene pi bond, and then nitrogen gets its lone pair back. And so formally, that's the production of H2O water. So you can form the E and Z isomers from the alpha prime. If we did the elimination from the other alpha carbon, we just get a situation of that alkene, because the remaining two substituents are both hydrogen, there's no E and Z possible. Questions? So in the lab, chemically, that's how these are formed. Imines and enamines are important functional groups for biochemistry because a resonance form of that, you can push that lone pair of electrons back down here. And in biological systems, most carbon-carbon bonds are formed using enamines. And again, that's a preview of a later module. So we have this ketone. We have this secondary amine. What do you think the product is? So yeah, B is the correct product. So without spending your early to mid 20s writing out the mechanism of this, what's a way to kind of figure things out. So that's oxidation state 2. This is an alpha carbon. For an enamine, the double bond is always going to be between that. And then off that oxidation state 2 carbon is going to be whatever your amine is minus the hydrogen.
Wikipedia.
Series been invoked. So you're just making that sort of connection. If you're able to sort of put those markers in the ketone or aldehyde, then form that carbon-nitrogen pi bond, that's really what you need to recognize without having to draw the mechanism each time. So let me see where we're at. So next week, basically, what we'll continue on with is we're going to use those imines and enamines to make higher order amines, because in the prior module, we only learned how to make primary amines. We can use those things now to make secondary and tertiary amines.